Alrighty, we should be able to come back on stage here. All right, so we still have about 8 minutes before we start. I do see we have Pradeep has joined us in the audience. Hi, Pradeep. Good morning. I guess good morning for me. You're good evening where you are, I think. But yeah, Michelle, if you wanted to share, we can still try you emailing me the slides if you want to just keep that as a backup. But it is your call.
Got it. Yes, I will send that over. I think my internet is still a little spotty, so if I'm unable to even send my slides, I will try to turn off my video, and hopefully that will be okay.
In the absolute worst case scenario, if you feel it's not going to work, we can reschedule to another time that works to your schedule. Because sometimes the internet is just doesn't work for us, and when it's beyond your control, there's nothing you can do like that.
Yeah, that's very unfortunate. I'm very sorry. Problems.
That's not a problem. Sometimes that just happens.
Okay, so I'm not sure if you can hear me well. I am having some trouble sending the slides, but I'm going to share my screen and see if voice significantly cuts out a bunch. So how is my audio?
You're coming through fine just now. Sorry about that. I'm just recovering from a cough, and I still have a raspy throat every now and then. Would you mind trying to just talk while changing slides just to see? Because that's when it's going to process the most.
Yes, so, okay, changing slides here, changing slides now.
Yeah,
How
that's
is
coming through
this?
fine.
How is my audio? Okay, great.
I think you're set. It's working.
It's great.
Okay.
There's one
Awesome.
question. There are people waiting to enter. Should they register? Should they use the magic link? Because I'm receiving some messages now.
Sorry, what was that, Alexandros?
I am receiving some messages from people that they are trying to connect, and what they see is just the screen of the ISCB Academy. Should they use the magic link to enter the event?
Yes, yeah, you have to log in
Okay.
to Nucleus to get there. So you have to use the magic link through the login in the top right.
Okay.
Once they're in, it should once they've logged in, it should automatically load them in here. If it doesn't, refresh the browser and it should bring them.
Great.
And if that doesn't work, send them to me and I will get them sorted.
Okay.
All right, so we do have some people joining us now. We are at the start time. So Alexandros, if you want to take it away, we can get going here.
Okay, thanks for hosting that, Seth. Hello, everyone. Today we're excited to host a webinar of the ISCBacademy of NetBio COSI entitled Contextualizing Protein Representation Using Deep Learning on Protein Networks and Single Cell Data. area aims to model gene pleiotropy by generating different protein representations depending on the cellular context. Our invite speaker for today is Michelle Lee, a last year PhD student on the Bioinformatics and Integrated Genomics program at Harvard Medical School. She received her Bachelor in Mathematics and Computational Science from Stanford University, where she developed computational methods to study the mechanism of antimicrobial resistance and susceptibility. At Harvard, she continues to design novel machine learning methods to expedite diagnostic and therapeutic endeavors. The overarching objective of Michelle's research is to generate contextualized representation for hard to diagnose and hard to treat patients. Today, she will walk us through some of her most recent work. With no further delay, Michelle, the stage is yours.
Thank you so much, Alessandros, and thank you, Seth, for this incredible opportunity, both you and Alessandros and the ISCB community. So hello, everyone. My name is Michelle Lee, and I am really excited for this opportunity to share some of my PhD research with you. So today, I will be sharing about contextualized protein representations, learning on protein network and single-cell data. So, what is context and why do we need it? So let's take Apple, for example. If we were to use it in, would we get the same recommendation GPT or the like? Well, no, because Apple means different things depending on its sentence. Now, the same is true in biology. H2AFX is a gene whose function or role depends on its context. It has many different functions depending on the cell context. Because of that, gene expression and protein function depend on cellular and tissue contexts, and biological functions can vary considerably across healthy and disease states. Models that aim to generate predictions on proteins should consider the different types of contexts that proteins may operate under. But current protein representation learning approaches, including those that are structure or sequence-based, are context-free, as in each protein has only one representation learned from a single context or an integrated view across many contexts. And these context-free models are unable to dynamically adjust outputs based on GNN network for protein networks and biology. I'll discuss the data that we're using, and then I will describe the detail. So, to be able to model proteins across cell type contexts, we need cell type specific protein interaction networks, which are the networks in the peripheral. So, to construct these cell type specific protein interaction networks, we start from a multi-organ single cell transcriptomic atlas of humans, which consists of samples from 15 donors, resulting in almost 500,000 cells after quality control processing. For each cell type, which are classified by this atlas, we get a list of genes that are activated in the cell type. In short, activated means that these genes are a higher average expression in the cells of interest, aka the cells that are classified as a given cell type, compared to the rest of the cells. And from this list of genes, I take the global reference protein interaction network, extract the proteins corresponding to the activated genes, and take the largest connected component. As a result, each cell type has they stay.
Michelle, we cannot hear you very well now.
Incorporates application and tissue. How's my questions? Are you better?
Can you repeat a bit this part of the slide, maybe, because it wasn't very clear, the sound.
About organization. exactly kind of overlaps. Here we have our cell type communication. Yes. Okay. All right, I will repeat this slide. So first, we have our cell type node, these diagonal lines. The colors represent cell type identity. And these cell types are given by tabula sapiens. We also have tissue nodes. The size of the tissue nodes indicates the level in the hierarchy. So the smaller the node, the closer to the root node of the ontology. Each of these cell types come from a specific tissue according to tabula sapiens metadata. And so we draw edges between those tissue nodes and the 156 cell type nodes. Cell type to cell type edges mean that the cell types interact, and this is based on their ligand receptor expression. So we select ligand receptor interactions that have a p-value of less than 0.001. And finally, we have our tissue-tissue relationships that are tissue relationships. So we take the nodes corresponding to the tissues that the samples are the parent. Our data model. So let me first get into that. So given our cell type specific networks, Pinnacle embeds proteins, cell types, and tissue nodes such that proteins from the same cell type network are attracted to each other if they have an edge. And by attracted, I mean their embedding distance is minimized, is shorter if they have an edge between them, and larger if there is not an edge between them. Protein embeddings from the same cell type network are attracted or closer to the embedding of the corresponding cell type and repelled from other cell type embeddings that cell type does not belong, that those protein embeddings do not. Similarly, type and tissue nodes that share an edge are encouraged to be embedded close by, and those that do not share an edge are encouraged to be farther apart, and so on and so forth. As a result, we have a single unified embedding space where protein representations are organized according to cell type context and tissue organization. In these UMAP plots, notice that the proteins from the same cell type context are enriched in a particular embedding region. And we actually use an approach called SAFE, called Spatial Analysis of Functional Enrichment, to show that indeed the spatial regions in Pinnacle's embedding space are significantly enriched for protein representations originating from the same cell type context. Okay, so let's model the report F. So first, how do we learn embeddings of each node? There are different levels of learning here. There is protein level, type level, and we use something called an attention mechanism. An attention mechanism as what would you pay attention to? So let's say this I is your mom. So, and a book. Probably, and that's the least to need coffee to see is applied for proteins. So,
Michelle, sorry for interrupting. If you could repeat this part again because it's breaking.
Yes. So to briefly repeat, so the first part I'm explaining here is the intuition behind an attention mechanism. So when you're trying to, when you think about attention mechanism, how or what would you pay attention to the most to achieve the goal? So if this eye is your model, that's your objective. You have 5 items to choose. So our options are newspaper, blank paper, coffee, notebook, and a book. We'll probably pay the most attention to the book and the newspaper, and probably the least to coffee, unless you need coffee to stay awake to read your story. But the same idea is applied for proteins, cell types, and tissues. So here we are to learn an embedding of this protein. We want to pay attention to say this protein the most, this protein neighbor the most, and then this one and this one. For our cell type embedding, so we can maybe pay the most attention to this tissue neighbor, this cell type neighbor, and then these two might not be as important, and so on. There's also this attention bridge here that is essentially allowing us to propagate information from proteins to cell types. So we have 156 networks and 1 metagraph. These are disconnected, which does not bode well for graph neural networks. We assume that the networks are connected in some way, or at least that's ideal for learning and embedding. So we need a bridge between these disconnected networks, and that is exactly what the attention bridge is aiming to do. So for each cell type embedding, which protein embeddings should be most important for learning that cell type embedding? So you can think of this as a way to learn the important protein biomarkers for a cell type automatically, and the vice versa. Ways to then learn the protein embeddings. I can have multiple let's go into the math. Let's take a look at some things on how embeddings and how object optimized. First is our team attention with cell type specificity. EP means we are dealing with protein. So again, P is protein. And for each, we are aggregating nodes and variable. Is our protein edge type matrix to project the features of the protein in its cell type specific PPI network. And there is one unique weight matrix for each. And so essentially, you can think of protein H is an attribute of neighbors learned. The similar idea is true. So every cell type name are aggregate the neighbors embeddings given with some attention mechanism and the transformation matrix that is specific to cell edges. Then we do the same before cell type to tissue, and we aggregate that so that we can type edges and cell type to tissue edges separately. And the analogous is true for tissue to tissue, tissue to cell type, in order to learn tissue embeddings. And finally, this protein to cell type bridge that I was talking about. So in order to learn our cell type embedding, we aggregate the proteins that are activated in that cell type. So the embeddings of the proteins activated in that cell type, and we learn an attention mechanism to weigh the importance of each of these proteins. And the vice versa is true. So when we are learning protein embeddings, we use those same weights to weigh our objective has 3 components. It has a loss at a protein level, at a cell type level, at a tissue level. At the protein level, we perform link prediction on protein-to-protein interactions. We also predict which cell type the tissue is activated in. At the cell type level, we perform link prediction, and we predict cell type to cell type edges and cell type to tissue edges. Similarly, at the tissue level, we predict link prediction at the tissue-tissue level, for tissue-tissue edges and for tissue cell type edges. So we use binary cross entropy for the link prediction. And cell type classification, we use center of loss. And what that essentially means is you try to minimize the distance between your protein embedding and its corresponding cell types embedding. So basically, you want your protein embeddings in a certain cell type to be closer to the cell type embedding. Okay. So altogether, Pinnacle provides a set of contextualized protein representations, cell type representations, and tissue representations. In contrast, prior methods only provide one embedding per protein. So with Pinnacle and the protein representations, we are able to perform multimodal by integrating diverse contexts into a single contextualized model, transfer learning across contexts, learning a single unified embedding space where cell type specific protein activity, cell type communication, and tissue context are all used to generate contextualized protein embeddings and generate contextualized representations. Okay, before I move on to results, do we have any questions about the approach?
I think, Michelle, at some point, someone in the chat was asking how you're creating the cell type specific PPIs because it was breaking. So if you could, like in a nutshell, repeat that part, maybe.
Yes. Okay. So to summarize, we have our single cell transcriptomic atlas on the left here, and we extract the activated genes in these cell types. And by that is that these genes have a higher average expression in our cells of interest compared to the rest of the cells. And of course, you can use other metrics as well to define activated cell type genes. With this list of activated genes, you go into your global reference PPI network and directly extract the induced subgraph of those activated genes. And that becomes, when you take the largest connected component, becomes our cell type specific PPI network. Of course, you can use other approaches to construct your cell type specific PPI network. But for our purposes, we went with the most conservative approach. Is that more clear?
Yeah, the sound was perfect now. Thanks for repeating the slide again.
Okay.
Thanks a lot.
Yes, thank you for, yes, please, at any point, go on, please. I appreciate you letting me know when my sound cuts out. Okay, so revealing my, okay, there you go. All right, any other questions before I move on to results? Okay, so let's take a look at our results.
In the Q&A, there's a question,
yes.
Michelle, about if you could repeat a little bit how attention was used for creating the embeddings.
Yes, I can. So, yes. So you can think of attention. So this is, if you're familiar with graph attention neural networks, it works similarly. So essentially, let's just take proteins, for example. So for every node, you're going to have an embedding that we continuously update during training, right? There's also we incorporate a edge type specific weight matrix, transformation matrix. This is quite standard for graph convolutional networks. But the part that we care about here, that you're asking about, is attention weights. So these attention weights alpha is essentially the weight between node U and node V. And essentially, without these attention weights, it's a naive average, an unweighted average of the embeddings of the neighbors. With these attention weights, then it's a weighted average of the neighboring embeddings. Is that a bit more clear? Okay. Thank you. Thank you for that response. Okay, great. Any other questions? All right, I'm not seeing anything, so I will move on to results. Okay. Okay, I hope that my audio is stabilizing. Okay, there you go. So let's take a quick look at Pinnacle's contextualized representations. So first, we want to know how well separated the protein representations are based on context. So the x-axis here is a similarity of Pinnacle's protein representations. And the y-axis is the compartment of each cell type. For example, we have epithelial cell types, immune cell types, endothelial cell types, and stromal cell types. And these are annotated by tabula sapiens. And I only use them for the purposes of having a relatively clean stratified analysis. Of course, we don't have to do it this way. But as you can see, so actually the dark dots here and the dark box here, they are the similarities of proteins from the same cell type. The light purple are similarities of proteins from different cell types. And as you can see, there's a significant gap between the dark and light purple similarities, suggesting that the protein representations are well separated according to context. When we remove the cell type and tissue context from Pinnacle in an ablation study, we find that there is no longer this separation of protein representations according to context. We also examine the tissue representations produced by Pinnacle. So the x-axis is the tissue ontology distance. So given a pair of tissue nodes, calculate the shortest path length to its lowest common ancestor. And the y-axis is the tissue embedding distance. So given that same pair of tissue nodes, calculate the cosine distance of their embeddings. And we expect a positive correlation. So the farther apart the nodes are according to the tissue hierarchy, the more dissimilar the tissue representations should be. But we don't expect a correlation coefficient of 1 because the tissue representations actually capture additional information that's not present in the tissue ontology. And as hypothesized, embedding distances in the latent space and the corresponding distance in the tissue ontology are positively correlated. In contrast, when you shuffle the tissue ontology, the correlation with distances and in the latent space is lost, indicating that proteins' tissue representations meaningfully capture the tissue organization. And since tissue organization is propagated throughout the cell type and protein embeddings of Pinnacle, all of Pinnacle's representations capture tissue organization. Okay, so now what? We have contextualized protein representations generated from our cell type specific protein interaction networks. What do we do with them? So given our contextualized protein representations, we can fine-tune them for any downstream task of interest. In this case, we're interested in predicting whether a given protein is a strong candidate for a therapeutic area of interest. This is actually a binary classification. So given a protein, is it a strong candidate? But because we have unique protein representations per cell type context, we can additionally gain insights about which cell types are highly predictive to examine therapeutic targets. So how do we define therapeutic targets? Sorry, protein targets. First, we have a universe of druggable proteins in our protein-protein interaction networks. These are proteins that are nominal targets of existing drugs. A subset of those proteins are proteins that have been studied in our therapeutic area. A subset of those have been investigated in clinical trials. And a subset of those have been investigated in clinical trials that have at least completed phase 2. So this becomes our positive training examples, as in they have a label of 1. This subset of druggable proteins that have not been studied in our therapeutic area of interest becomes our negative training examples. So the 2 therapeutic areas that we choose to study are rheumatoid arthritis and inflammatory bowel diseases. The numbers here are the positive examples per cell type specific PPI network. So how do we get here? First, we specify a therapeutic area, in this case, RA or IBD. Since there are subcategories of RA and IBD, we include those diseases in our broad definition of RA and IBD. Next, we curate clinical trials of these diseases and subset to those that have at least completed phase 2. And so these sets of proteins become our positive training examples. So when we fine-tune Pinnacle's contextualized representations to generate contextualized predictions, we find something incredibly exciting. And it is that Pinnacle's representations predict drug targets for RA and IBD significantly better than context-free methods, and that Pinnacle is able to find the most predictive cell type contexts. So on the x-axis is our performance metric, and y-axis is our evaluation of the different models. So the first row is Pinnacle. Each dot here represents a cell type context. The next 3 are baseline models, all of which are context-free. Bionic is a set of graph convolution networks that integrates cell type specific protein interaction networks for each protein. GATT is a graph attention neural network that learns protein representations from the global reference PPI network. And random walk is a diffusion-based approach that operates on the global reference interaction network. Notice that for the RA and IBD model, there are many cell type contexts that outperform the baseline model. So the higher, the better. And let's just take a look at what these cell types are. Why are there cell types that perform better than these baseline models? And why are there cell types that don't perform as well as these baseline models? So here is our model using metric average precision and recall at 5. So each dot represents the performance of proteins from a single cell type. The lines here are baseline model performance. So this is bionic. This is graph attention neural network, or GATT. Again, these are cell type agnostic. As you can see, there are many cell types that outperform the global reference, the GATT model and bionic, which suggests that studying the therapeutic potential of proteins may be more accurate when leveraging the cell type context of those proteins. And these rankings have been validated by an RA clinician as being appropriate and very exciting hypotheses produced by Pinnacle. We can also look into individual proteins that are already targets of drugs and examine the most and least predictive cell types to better understand their mechanism of action. Here we have IL-6R, which is a very commonly targeted protein. And we have validated that these top 5 most predictive cell types, and one particularly exciting cell type, actually I did not list that here, my apologies. But these top 5 most predictive cell types are actually very exciting and validated by experimental results. And so our collaborator and us are looking more closely at these predictions now. We can also look at IBD, and we find that similar results are true. So the top most predictive cell types that outperform the baseline models are also highly relevant, highly predictive, and relevant to the literature for IBD. And we have worked with a clinician from MGH, Massachusetts General Hospital, to verify our findings. Again, we can also look at the protein level and the cell types that are predictive and least predictive for studying this protein for IBD. Okay, so now another important aspect of therapeutics is predicting protein-protein interactions. For example, in immuno-oncology, interactions like PD-1 and PD-L1, CTLA-4 and B7-1 are key players in cancer immunotherapies. However, our 3D molecular models of these proteins are not context-specific. So we're curious whether injecting context using our contextualized protein representations would help us better differentiate between binding and non-binding proteins. So we first take the protein representation learned from 3D protein structure and concatenate it with either a context-free embedding or a context-aware embedding. So the context-free, or the no cell type context here, embedding, that is by taking the average of Pinnacle's context-aware embeddings. So basically averaging out the context. Here, the cell type context is taking Pinnacle's contextualized representations. And we indeed find that contextualized representations allow us to better differentiate between binding and non-binding proteins. So on the left are results from context-free representations And on the right are results from contextualized representations. So the black and purple dots represent the average score of the binding proteins. And the gray and light purple dots are the scores for the non-binding proteins. And score here means cosine similarity between the embeddings. So the important comparison here is the gap between the binding and non-binding proteins. So with the free representations, the gap is very small. It's from 0.003 to 0.006, which you can argue is no gap at all. And the context-aware embeddings give us larger gaps between 0.011 and 0.015. Yes, the differences are still quite small. We actually find that they are statistically significant. So there is a lot of potential to incorporating context to generate contextualized predictions for precision medicine. So let's imagine some scenarios together. So given a patient with a loss of function and mutation G had an understanding of that gene's cell type specific activity, the recommendation may be to consider compounds whose mechanism of action regulates gene G in cell type C. And that requires us to have our cell graph as well as contextualized AI model. We also have another example where given a patient with certain symptoms and taking certain medications, if we had an understanding of those phenotypes and medications context in biology and medicine, then we could identify a diagnosis based on the diagnosis of other patients who have similar profiles. And it would be helpful as well to know what kind of lab tests are needed to confirm this diagnosis. And that is with the help of our medical knowledge graph and contextual AI method. So let me wrap up a bit with our key takeaways, and then I will show you a demo of Pinnacle. So Pinnacle is a flexible geometric deep learning approach that uses contextualized networks to generate context-aware representations. Here, we create contextualized protein interaction networks using single-cell gene expression to help resolve protein interactions with single-cell resolution. And Pinnacle does this by learning contextualized protein interaction networks, learning on these contextualized protein interaction networks to generate context-aware protein representations based on cell type activation. Pinnacle representations can then be used for downstream tasks that have cell type dependencies or actions of mechanisms of action. For example, nominating therapeutic targets. Now, with contextual learning, as we show by Pinnacle's performance, we can actually improve the accuracy of predicting drug specificity, safety, and toxicity, and of patients' response to treatments with cell type dependencies in the near future. All right, so assuming our internet permits, let me share a different screen where I can demo a little bit about Pinnacle. And in case my internet cuts out for whatever reason, here is actually a demo that you can open up on your own screen. Okay, so I will share another window. You should see a Hugging Face space where the title is Contextualizing Protein Representations with Pinnacle. All right, so if I scroll down, I can actually use different kind of settings here. We can look at the protein level predictions and representations, or the cell type level. Let's start with the protein level. So let's say I'm interested in TNF, looking at the different contexts of the protein TNF. And we can submit. Hopefully my internet allows us to process this relatively quickly. And okay, great. Now, hopefully we can see our embedding space. The different proteins in our embedding space are in gray here, and a star are the instances of our TNF protein. All right, that's great and all, but now what? Let's take a look at some more details here. So we can actually, well, we have the coordinates of these proteins in different cell type contexts that can hopefully help us identify the different cell type specific TNF representations that we might care about. We can also look at the cell type view, where we can examine a specific cell type of interest, let's say a B cell. And we can look at the proteins that are activated in that B cell, in B cells, and other information that are provided. So for example, here are in purple, the embeddings, the protein representations of proteins that are activated in B cells. You can also add other types of cell types and look at them on top of each other. Okay, and there are also other statistics that may be helpful. For example, well, the number of proteins are in these networks and our safe scores that I talked about are statistical analyses of spatial enrichment for these protein embeddings given the cell type context. Okay. Awesome. So now I'm very open to questions, anything that you want me to repeat or to elaborate on. Thank you so much.
Thanks for the great presentation, Michelle. If the audience has any questions, please pose them in the Q&A part of the chat. Maybe until the audience have any questions, I can ask one small question. So what are the contexts you can mainly integrate in your method, and how scalable is the method to different contexts?
Yes.
Like, is there on the context you can integrate, or it can go like beyond?
Yes, absolutely. So yeah. Awesome. So yeah, that's a really great question and really important question. So we are actually having follow-up studies. Actually, hold on. Before I continue, can you hear me well?
Yes, that sounds great.
Okay, great. Okay, so yes, so we are actually working on several follow-ups where we are now scaling our approach pinnacle to much larger atlases. For example, atlas that you may be familiar with by the Chan-Zuckerberg initiative, and we are working on scaling it from 156, which we see here, to even like 500 or more cell type contexts. So we are very excited about this kind of follow-up study, and hopefully we can scale it such that you can use it for any particular context that you're interested in integrating or using. In terms of what kind of cell type contexts, as I want to also add that you're not necessarily limited to cell type contexts or at least atlases of healthy individuals. You can also for the RA and IBD specific tasks, you can certainly integrate IBD specific atlases, as in transcriptomic data taken from patients with IBD, similarly for RA. So then you can have a more, RA or IBD or task-specific contextualized networks and protein embeddings for you to make to use for downstream prediction.
Yeah, that makes a lot of sense. So there's also a lot of direction, precision medicine, therapeutics, a lot of different areas that these models can be applied in general. That's thanks for the answer. That's great.
Yeah, thank you.
I don't see any questions in the Q&A. Oh, there is one question.
Okay. Do you consider including other omics data in the integration? Yeah, absolutely. So you can certainly do so. So one could consider integrating data like protein abundances. If you have information about that, and that we know that correlates a bit better with protein-protein interaction than transcriptomic data. Certainly, that is possible to integrate into our construction of the context-specific networks. So any sort of data, we are just showing here single cell transcriptomic data, single cell RNA-seq, just because we have an abundance of that, of those types of atlases. But certainly other modalities would be really important and very, very easy to integrate into our approach.
Any other question, comment, or something? If there are not any other questions, maybe we can wrap it up here. Thanks again, Michelle, for the very nice talk and introduction to this fascinating field of research. Good luck with your workshop in EURIPS on Friday. And thanks again for the talk.
Yes, thank you so much. And I do apologize for the internet connection problems. But before I close, if I may add some links that I included in my slides. So then if you're interested, you can look at the website and the demo for more information.
Okay, great for sharing.
Great, thank you so much.
Have a good day.
You too.